So in our third stage of translation, when we have gone over stage 1, which was amino acylation of tRNA, activating that tRNA by adding in that amino acid, then we went over initiation, which was bringing that AUG, the start amino acid, which was N-formyl methionine in prokaryotes, just methionine in eukaryotes, and binding that to the P site. But now we have the A site and the E site. They're still empty. The mRNA is bound, and we have the full ribosomal structure. But we have to keep on growing this peptide. We only have one amino acid right now, but a peptide or a protein is made up of multiple amino acids. So how do we do that? Well, we need to keep adding on amino acids. So essentially, how do we do that? Well, we have our mRNA bound, so we have to keep on reading this code. We've only read AUG. We have only read one codon. We have to keep on reading the mRNA, and we keep reading each codon, and each codon corresponds to an anticodon, and each anticodon corresponds to an amino acid. So let's say we move on forward and we read the codon that is over here. We're going to bring in a tRNA with the anticodon, and that tRNA is going to be carrying an amino acid. The amino acid corresponds to whatever the codon is over here. But what's interesting is that this tRNA, it's coming into the A site. And when it comes into the A site, it has this elongation factor bound. It's important to understand that during the initiation stage, we had initiation factors. But during the elongation stage, we're going to have elongation factors. So our first elongation factor is called thermounstable, TU. Thermounstable is bound to a GTP. And this thermounstable elongation factor bound with a GTP is going to bind with the tRNA, with the amino acid bound, and it brings it into the A site. When it brings it into the A site, the whole job of this elongation factor is to bring it into the A site. Now it must dissociate off. It dissociates off, but as it does so, we see the hydrolysis of GTP into GDP and inorganic phosphate. So the energy of this hydrolysis catalyzes this reaction. But now we have thermounstable with GDP bound. In order for this cycle to occur again, we need to attach a GTP to the thermounstable. So how do we regenerate that? First, we need to remove the GDP on thermounstable. We do that with the help of another elongation factor called thermostable. Thermostable comes in and it removes that GDP and it binds with the thermounstable, and we get thermounstable and thermostable bound together. When they are bound together, now the thermostable can help facilitate the bond with GTP and thermounstable. So GTP will now come in and bind to the thermostable and the thermounstable, and the thermostable will now leave. Now that we have a elongation factor thermounstable bound to GTP, it can now bind to a tRNA. When it binds to a tRNA, we regenerate this structure over here where we have tRNA bound to a elongation factor TU-GTP, and then once again we can bring it into the A site. So this is essentially a cycle where this is our end goal. We want this because this is going to facilitate bringing it into the A site, and then the cleavage of this GTP allows for this cycle to continue. And the whole purpose of thermostable is to allow thermounstable GDP to be converted to thermounstable GTP. So once again, these two, TU and TS, are elongation factors. Now we have a tRNA in the A site, and we have a tRNA in the P site. The P site contains the start amino acid N-formyl methionine with the codon AUG and the anticodon UAC. We need to move this N-formyl methionine to this amino acid. So how do we get this F met to go to this amino acid, whatever it may be, whichever amino acid that is. Well, on this amino acid, for an amino acid in general, we have an alpha carbon, we have a carboxyl group, we have an hydrogen, we have an amino group, and we have some sort of an R group. The amino group on this amino acid is going to nucleophilically attack the carboxyl group on this amino acid. And when it does so, we form a bond between these two amino acids. Methionine is an amino acid, so essentially one more time, this amino acid has this amino group, and this amino group nucleophilically attacks that amino group. So essentially this amino acid will move over to this side. And that's what happens. So now we have these two amino acids bound together, and it's in the A site. This tRNA no longer carries an amino acid, so it has to be shifted over to the E site so it can exit. But how do we cause this shift? This shift will require another elongation factor. Elongation factor EFG, which has a GTP bound to it. This elongation factor is going to bind to the A site. When it binds to the A site and we undergo the hydrolysis of this GTP, it will cause a shift in this way. So the tRNA that was in the P site gets moved to the E site, and this peptidyl chain that was in the A site gets moved to the P site. And we can see we underwent the hydrolysis, we lost a phosphate. Now this elongation factor has a GDP bound to it. And we're going to remove that now because we no longer need it. The whole purpose of adding the elongation factor with a GTP bound was to create that hydrolysis energy which was going to shift everything over this way. Now this tRNA that's in the E site is going to exit, and our growing peptidyl chain is in the P site. And now we are ready to bring in another tRNA. Remember, how are we going to bring in that tRNA? Well, we're going to have a tRNA bound to the elongation factor thermo-unstable. Thermo-unstable has a GTP bound to it. So thermo-unstable, it has GTP bound to it, and it's going to bind to a tRNA. So this is our tRNA. It's carrying an amino acid. Thermo-unstable is bound. GTP is bound. It's going to come into the A site. It's going to come into the A site. Remember, thermo-unstable, we're going to regenerate it with the help of TS. And then once again, the amino acid on this one, that's going to be right over here, the amino group is going to nucleophilically attack the carboxyl group on this amino acid. So then this is going to shift over this way. And the process will keep on occurring until we reach a stop codon. A stop codon can either be, the way we can remember it is we write out U, and then we say U, ga, ga. These are our stop codons, and when we reach a stop codon, we're gonna undergo stage 4 of translation, which is termination. But for now, all we need to understand for elongation is that for every amino acid that we bring in, we're going to require a GTP right over here, and we're going to require a GTP right over here. So elongation requires 2 ATP equivalents. And that's elongation. It's going to keep on happening until we get that peptide and we reach a stop codon. And the mRNA is going to be read from the 5' to 3' direction. And each codon contains 3 nucleotides, for example, AUG, which has a complementary anticodon. In this case, it's UAC. And that anticodon on the tRNA is carrying our desired amino acid.